Thank you everyone for joining us today for the second webinar in our four-part series on science and risk-based stability practices in pharmaceutical development. Our topic today will be on the application of science and risk-based predictive stability to biological therapeutics. All of our speakers today work with the IQ Consortium Science and Risk-Based Stability Working Group and share an interest in the using this predictive techniques we will talk about today, as well as other science and risk-based stability approaches to accelerate product development and focus stability knowledge and use throughout the product lifecycle. Lori McCaig is a Director of Stability Program Strategy at CGen, responsible for standards, governance, and innovation across stability programs throughout the product lifecycle. Before joining CGen, she led global stability program management at Genentech Roche, and prior to that held leadership positions in stability, lab, and quality management at a number of companies. In addition to working with the IQ Working Group, she's served on the USP Biologic Stability Expert Panel and Physical Stability Subcommittees, as well as serving as the Vice Chair of the AAPS Stability Community and working with the ICH training partner at Northeastern University, developing and presenting ICH Q1 stability training to global health authorities. Andrew Leonard is in Amgen's external engagement advocacy team within CMC Reg Affairs. He has 15 years of CMC regulatory experience, mostly focused on the EU for clinical trials, marketing applications, and post-approval variations. Before moving to regulatory affairs, he was a bench scientist in drug discovery for pharma biotech startup companies. He has a PhD in biochemistry from Cambridge University in the UK and is interested in applying prior knowledge and risk-based control strategies to biological medicinal products with a focus on stability, as well as introduction of new technologies to accelerate the design, manufacture, and control of biopharmaceutics. He actively represents Amgen in many FPA work streams and currently leads the FPA stability work stream, while also participating on the IQ Consortium stability activities and the bio team. Kayla Woodleaf is a Director of Stability and Data Analytics at Vir Biotechnology. She has over 15 years in industry experience in stability program management, ranging from first-in-human clinical studies to post-approval and lifecycle management. Prior to joining Vir, Kayla led the biologic stability team at Biogen, where she developed phase-appropriate stability strategies for clinical and commercial assets and leveraged risk-based predictive stability approaches for regulatory filings. In addition, she was responsible for the establishment and management of release and stability specifications. She also works with the ICH training partner at Northeastern University, developing and presenting training for global health authorities. Dan Willingmeyer leads the clinical large molecule stability and specifications team at Biogen. Before joining Biogen, Dan worked on the development of cell-based and high-throughput screening assays in the pharmaceutical industry. He has over 15 years of industry experience and currently specializes in managing large molecule stability strategy, specification setting, and leveraging prior knowledge approaches to support product development. And he participates in the IQ Consortium's risk-based predictive stability working group, focusing on justification of lean stability strategies. So with that introduction, I turn it over to Lori to kick off the webinar.
Thanks, Brian. I'm going to get us started, give a very general overview of science and risk-based stability approaches and predictive stability, and then Andrew, Kayla, and Dan are going to walk us through a few of these examples and what's next, and we'll end with some Q&A. So let's take a minute at first to talk about the IQ Science and Risk-Based Stability Work Group. Our mission is to advance the use and applications of science and risk-based stability approaches for pharmaceutical products, both small molecule and large molecule. We do this by working together to understand the current and emerging approaches and trends across companies and to encourage innovation. Publications and workshops such as this one take that conversation and that mission to a broader audience. So the work group then is through the month of February presenting this series of science and risk-based stability webinars every Thursday. Last week, we talked specifically about some of the technical predictive stability approaches to small molecule therapeutics, and today we're going to concentrate on predictive stability for biological therapeutics. I would throw in that they're often applicable in vaccines as well. The third week, we're going to talk about leveraging available knowledge to design appropriate stability studies, and we're going to end with week 4 on the regulatory landscape of science and risk-based stability, both for clinical development phase, product registration, and post-approval changes. So what exactly is science and risk-based stability? It's not just the long-term and accelerated stability data with a few special studies thrown in that has long provided evidence for product stability and shelf life, but it expands that. It's about using the knowledge space created by the various inputs shown on the right here, combined with incorporation of risk assessments, both formal and informal, for stability risk, in order to, one might put it, know what we know and focus on what is important, and have that play a greater role in the design of our stability strategy and the stability data packages. This can be as simple as lean stability protocols, where you reduce the number of time points or attributes tested on your commercial stability, but it's more than that as well. These new approaches provide a scientific basis to the evaluation and use of stability data and are consistent with the more recent ICH guidelines for science and risk-based approaches. They do require a fresh look at the ICH stability guidelines, which were developed earlier and are not as explicit about science and risk-based approaches. It's important to note that while these approaches might provide a balance, such as the amount of stability data provided at submission versus what is provided later, they cannot and do not compromise the product quality or patient safety. In fact, and in particular, the predictive approaches that we're going to talk about today, which are bolded here below, do provide that assurance, just in a different way for stability. So in addition to what is science and risk-based stability, it's also good to think about where it can be applied. The needs for stability understanding and stability data change throughout the development cycle and commercial life cycle. Traditional long-term stability, such as one would find in formal stability study designs listed here, is not necessarily the best tool for every scenario at every stage. Predictive stability models can provide a real advantage and accelerate the timelines at several key points in the product life cycle, from formulation to getting the product into the clinic, to the approved product to the patient, and through the post-approval life cycle. So that brings us to the key takeaways from webinar 2. What we're going to focus on today are the application of science and risk-based predictive stability to biologics. And this predictive stability is a subset of the overall concepts of science and risk-based stability. Two approaches being developed for biologics that we're going to focus on are prior knowledge from justified-like molecules and empirical kinetic modeling. Both of these approaches provide accurate inference of product quality, attribute, stability profile, and setting of shelf life. And we'll talk about some of those model choices and applications of where they fit as well. So in beginning our discussion of science and risk-based predictive stability, let's get back to that traditional linear development, where from the start to the launch, it can take up to 12 years. And real-time stability data generation is needed at several points within this process here, leading to a timeline that is not aligned with today's accelerated pathways. In particular, the time generated to support product into the clinic, and then the time needed for that long-term data generation to justify a commercial shelf life that's practical for supply chain, allows usage of those PPQ type lots, and satisfy the requirements for remaining shelf life in many regions. These are sticking points, and these are places where that predictive stability, which can give you the answer of product stability and shelf life in a shorter period of time, can really facilitate earlier access to new medicines. So do we just apply the same types of predictive approaches that were talked about last week in the small molecule focus? Can these same models be applied to biological products? And the answer is not really. They're not often immediately applicable. There are unique challenges for biological products, and these result from the microheterogeneity, from biosynthesis to the potential post-translational modifications, for example. The combination of chemical reactions and that impact on biophysical interactions, some of which may be reversible, can lead to very complex kinetics for those large biologics compared to the small synthetic molecules. There also is a real concern about the interpretation of certain stress stability temperature results whenever they're near transition temperatures. An example is the glass transition temperature for our lyophilized product, where that degradation is less stable or predictable. So the short answer is that new predictive approaches needed to be designed to apply to biological products. So at this point, I'm going to hand over to Andrew, and he's going to provide us some examples of how that's done.
Right, next slide, please, and thank you, Laurie. Now, in recent years, two main approaches to predictive stability have been developed for biological therapeutics. One requires the sponsor to have sufficient experience in manufacturing a particular structural class of biological therapeutic, often referred to as a modality, and thereby have adequate transferable stability knowledge of these molecules to apply as prior knowledge to a new molecule under development. The other approach is kinetic analysis, which you have heard about in webinar 1 of this series for ASAP, is derived from Arrhenius equation and brings together stability data obtained at different temperatures. So I'll be discussing both biological therapeutic predictive stability approaches, starting with kinetic analysis. So the degradation pathways for biological therapeutics can be complex compared to small molecules, with high-order nonlinear reaction kinetics. First-order reactions may not predict the long-term stability data for all quality attributes. Indeed, interactions between quality attributes can lead to complex two-step reactions, such as chemical modification changing the protein tertiary structure, such that the formation of another modification is accelerated. Therefore, an empirical approach has been taken, where stability data are fitted by software to kinetic equations. The best fit equation is then used to extrapolate the long-term stability profile. And advanced kinetic models that have been derived from Arrhenius equation have recently found to be suitable for complex biologic quality attributes and successfully used in the evaluation of stability data for vaccines and therapeutics during the current COVID pandemic. However, it should be acknowledged that some biological therapeutic quality attributes for monoclonal antibodies have been found to be amenable to simple first-order Arrhenius analysis, and the investigators suggest that the attributes that don't fit the kinetics were not shelf life limiting, and that the approach could be used to determine product shelf life. So the slide you're seeing now introduces a more complex advanced kinetic analysis approach in which the stability data at different temperatures are fitted to increasingly complex kinetic models. The illustrated example from Klenny et al. Of Sanofi is for a recombinant protein antigen adsorbed to an aluminium salt adjuvant. Stability data through 6 months were obtained at 4 different temperatures for the shelf life limiting quality attribute. A best fit kinetic model was then selected to extrapolate the stability profiles. Other recommended storage condition, the extrapolated stability data were used to determine the proposed shelf life. Most importantly, the predictive stability model was verified by ongoing stability data and illustrates the accuracy that can be achieved using advanced kinetic modeling. This approach has now been successful for a variety of vaccine types and also for a COVID biologic therapeutic product. Next slide, please. Now, I'm sure that most of us that have seen stability data for a variety of different products of the same structural modality and pharmaceutical form have noticed that degradation pathways and kinetics are similar, and that the molecules are behaving as a class of product. Indeed, at Amgen, our extensive stability knowledge while developing IgG recombinant protein therapeutics and for the HLE BiTE modality provides the prior knowledge that supports the claim that these structural moieties can behave as a class. Clearly, by adopting platform approaches to a manufacturing process and analytical methods of control, one can cluster molecules according to their stability profile and justify the transfer of stability data across like molecules as prior knowledge. So as like molecules, I refer to different molecules that meet a set of criteria or considerations supporting the transfer of their stability data across a set of molecules that behave in with similar degradation pathways and kinetics as prior knowledge. Now, this prior knowledge approach assimilates all product-specific development stability data, and with those data from the like molecules. Next slide, please. So key to using prior knowledge is justifying the transferability of the data across molecules. We have addressed this by comparing a set of attributes that could potentially impact stability, such as the structural modality, manufacturing process, analytical methods, formulation, container closure, storage conditions, and the amount of stability data available. Importantly, it should be possible to justify differences and not having a meaningful impact on the stability profile. Therefore, these criteria define what is referred to as like molecules and indicate the degree of fit to a platform. Note that the regulatory filings using a prior knowledge approach, it is recommended to provide the comparison across the selected prior knowledge molecules in a tabular form, which can be part of the overall risk assessment needed in employing any science and risk-based approach in a stability program. So, next slide, please. So to generate a prior knowledge predictive stability model, we gathered a prior knowledge reference set of stability data from our most similar IgG products and also for the platform HLE BITE therapeutic modality. The molecules were selected based on the aforementioned set of criteria to justify the pooling of data from like molecules. These data were used to generate a stability profile model for the two therapeutic modalities, IgG and HLE BITE. Tolerance intervals were then calculated from the pooled data and plotted against the specification. For an impurity forming over time, the intersect of the upper tolerance interval with its specification denotes the shelf life for that attribute. The lowest attribute shelf life determines the maximum product shelf life that is supported by the prior knowledge approach. The data evaluation is essentially as described in ICH Q1E for extrapolation of stability data to determine expiry as applied to small molecules. However, we would not describe the prior knowledge approach as extrapolation of stability data, as it's the transposition of product-specific stability data onto a prior knowledge-derived stability model. So, next slide. Now I show some selected stability data reference sets for given product structural modalities that can be concluded to behave as a class, such that differences in amino acid primary sequence are not reflected in the stability profile of these modalities. So here we see 4 quality attributes for the drug substance of a product class that is stored frozen at temperatures below the glass transition point. Clearly, the stability profiles are flat for these attributes. Structural differences between the molecules result in time zero differences for some attributes, such as cation exchange acidic peak. However, it's the slopes that are important in evaluating a stability profile, and we can normalize the stability data, as done here for the cation exchange data. So we conclude that the stability profile for a frozen drug substance through 24 months is not time dependent, and the molecules behave as a class with comparable stability profiles. Statistical analyses support that there is no significant slope, and the tolerance intervals do not meet the stability limits. Therefore, a 24-month shelf life can be applied to like molecules that fit this frozen drug substance model. Next slide. So liquid drug products typically demonstrate some degree of stability indicating attributes that change over time, as exemplified by these IgG stability data. Here, cation exchange acidic peak, product fragmentation, and high molecular mass species all increase over time. However, the rates of degradation are slow, and the tolerance bounds remain within the assigned attribute stability limits. And the product data trend, within the time frame of the reference data set. So note that the intersection of the tolerance interval with the specification depends on the ability to gain agency approval for a more patient-centric approach in setting the specification. So a tighter limit reduces the time of intersection with the model tolerance bounds and reduces shelf life of that attribute, which may finally impact the final assigned product's shelf life. So this illustration uses stability limits that are considered safe for these attributes and were used in clinical studies. And in this case, a 36-month shelf life could be claimed for the investigated molecules that meet the model criteria. Next slide. This next set of data are for a drug product of a modality that is stored lyophilized and refrigerated. Again, this is a very stable storage condition with no significant time dependency for the stability indicating attributes, thereby allowing a 24-month shelf life to be claimed. Next slide. So another useful tool is the accelerated condition stability data. Although forced degradation may not always represent normal degradation pathways, accelerated conditions that don't cross phase boundaries, such as frozen to liquid, may be extrapolated at a high level to support the recommended storage condition degradation profile. Indeed, here we find that accelerated degradation can both identify non-fit molecules, as indicated here, and provide assurance that the model and investigated product are behaving as expected. So here we see stability data for 5 products in which one behaves atypically for an attribute. The slopes plotted on the right demonstrate that this product behaves differently to the other molecules, and this product wouldn't be included in the model. Furthermore, degradation pathway studies for this product did reveal the molecular root cause for the atypical behavior, and that a post-translational modification triggered accelerated formation of another impurity attribute. Well, that concludes my overview of kinetic analysis and prior knowledge approaches to predictive stability modeling. So now I'll pass you over to Kayla, who will further discuss the use of prior knowledge in setting shelf life for monoclonal antibodies and to support the end user time out of refrigeration condition.
Thank you, Andrew. And so today we'll discuss a case study to demonstrate using prior knowledge to support an expiry extension for a clinical product. Our prior knowledge data set consisted of approximately 120 drug product lots tested at release. Our stability data set consisted of 6 different monoclonal antibodies produced using our platform manufacturing process. Stability data were available for at least 24 months at the long-term storage condition and through 6 months at the accelerated and stress storage conditions. All samples were tested with our platform specification and stability indicating test panels. For all 6 monoclonal antibodies that are included in our prior knowledge data set, all of them were formulated at approximately 150 mg per ml, with an increase in high molecular weight species identified as our major route of degradation. As you can see at the long-term accelerated and stress storage conditions, we see very similar stability profiles for this product. Next slide, please, Lori. Now we will transition to utilizing this prior knowledge data set to assign clinical expiry to a new molecule named MABX. This was formulated also at 150 mg per ml and was introduced utilizing our platform. This included the manufacturing process, test methods, and specification. Approximately 6 months of data are available at the long-term accelerated and stress storage condition. In order to apply our previously established expiry of 24 months to this new monoclonal antibody, we developed inclusion criteria. This criteria consisted of a recommendation to have a minimum of 6 months of data. In addition, the release values for the new molecule should be within our historical prior knowledge range. Furthermore, we should expect to see a comparable stability profile. So as you can see here on the right, that we expect data generated on the new molecule to fall within our 95% confidence interval around our prior knowledge data set. In addition, we should also see consistent rates of change. Next slide, please, Lauren. And so you can see here in this table down below, our new molecule, MAVX, had very similar rates of change to our prior knowledge data set at both the accelerated and stress storage conditions. Furthermore, you see similar stability profiles with all data falling within our 95% confidence intervals at these storage conditions. Based on the conformance of this new molecule to our established model at all storage conditions, this allows us to assign the 24-month expiry to this new product. It should be noted that stability will continue to be monitored at the long-term storage condition to confirm conformance to our established model. While this was a very simplistic case with all formulations at 150 mg per ml, this approach could be applied to formulations with varying protein concentrations, different container closures, or other process changes, under the assumption that you are able to demonstrate comparability. Again, now I will turn it over to Dan Willingmeyer to provide further discussion on how you can use prior knowledge to establish timeout or refrigeration.
Okay, thank you, Kayla. So similar to how we applied prior knowledge to establish platform expiry in the previous case study, this new case study involves leveraging prior knowledge to support platform timeout of refrigeration allowances. These TOR allowances are required to support various operational steps during manufacturing and distribution, including visual inspection, device assembly, label and packaging, and as well as unintended temperature excursions that may occur during shipping and storage of drug product. In this case study, the example prior knowledge data set represents a wider range of formulations than seen in the previous case study and used in the platform expiry approach earlier. So this involved protein concentrations varying from 50 to 150 mg per ml, as well as different container closure systems involving vials and prefilled syringes. So the figure in this slide presents an overview of how the prior knowledge release and stability data were leveraged to support these TOR limits. It starts by leveraging prior knowledge release data to define the worst case release values for all attributes which demonstrate significant change as a function of storage time and temperature. In addition, prior knowledge stability data are leveraged to determine the worst case change due to the long-term condition through shelf life, as well as change due to the predefined TOR limits established in this process. These stability deltas are applied to the worst case release values and are assessed against platform specification limits. This allows us to demonstrate that under worst case conditions, exposure to the defined TOR limits will not adversely impact product quality or conformance to the stability specifications. Next slide, please. So here we show some of the analyses used in this process for analyzing the historical prior knowledge release and stability data. And this particular example deals with HMW by SEC. The colored symbols are representing different products in the prior knowledge data set. And having different products in this analysis can complicate leveraging statistics to calculate worst case release values. So we looked at several options, including minimum and maximum ranges, mean plus or minus 3 SD, and tolerance intervals in order to determine the worst case release value. So this value is important because it will anchor the subsequent calculations and also be used to define inclusion criteria for future programs to leverage these platform TOR allowances. There's also stability data leverage from long-term accelerated and stress conditions that were used in this assessment. And in the prior knowledge data set, we did see consistent stability profiles across these products. And as typically observed with HMW, there's a nonlinear increase observed at 5 and 25C with the early burst aggregation occurring, and therefore the square root of time transformation was applied to model these data. And the output of this is determining worst case rates of change within the prior knowledge data set, and that's shown on the bottom right. Next slide, please. So the next step in this process is to select the starting point to begin evaluating potential platform timeout of refrigeration limits. So this is a bit of a balancing act between what can be supported from a product quality perspective versus manufacturing process requirements and our expectations for control of product storage conditions. For example, we could have a product with a very robust stability profile that could be stored outside of refrigeration for months and not adversely impact conformance to specification. But these limits would not align with what is required or expected for manufacturing and storage of drug product. In this case, we looked at what was required during both manufacturing and distribution, as well as historical temperature excursions assessed for programs included in the prior knowledge data set. And through this assessment, defined initial starting TOR limits of 360 hours at temperatures between 8 to 25 C and 24 hours at 25 to 40 C. So the worst case value table here shows the impact of long-term storage and exposure to these TOR limits, which are calculated from the worst case slopes provided in the earlier slide and assessed against a release value of 1.5%. The analysis shows that these TOR limits will not impact the ability of drug product to conform to specification. A few things to note here is that this is a truly worst case assessment in several ways. Most importantly, that 25C stability data was leveraged to assess any excursion between 8 to 25C. And also we can note that most of the change occurring here is due to the long-term storage through 48 months, not necessarily the established TOR limits. So once this analysis is complete and we have assurance that all lots included in the prior knowledge data set can conform to this time out of refrigeration limit, these limits can be applied to new molecules as long as they are meeting some of these inclusion criteria, which include consistent storage conditions, methods, and specs to those used in the prior knowledge data set, as well as release values from the new product, which conform to the historical worst case values. With that, I'm going to hand it back to Leonard to discuss some new innovations in predictive stability.
Okay, thank you, Dan. So more recently, some companies have begun exploring the use of Bayesian statistics to incorporate prior knowledge into stability analyses, among other applications. Also, as biopharmaceuticals advance into the digital age, we're also looking into applying artificial intelligence, deep learning, to evaluate large stability data sets across a company's product portfolio, and to incorporate diverse sources of knowledge into a complete holistic evaluation of a product stability profile. Indeed, our Bayesian statistics analyses and use of artificial intelligence is helping elucidate which characteristics of a prior knowledge product really matter in determining a quality attribute stability profile. A third point to highlight is that although science and risk-based predictive stability has gained traction in the current pandemic crisis, the industry is advocating to agencies that the current enhanced understanding of biological therapeutics and their manufacturing processes should provide the confidence to expand the use of science and risk-based predictive stability from emergency access in a pandemic scenario to more generally products of unmet medical need and beyond. The tools are available for accurate prediction that could be verified even for biological therapeutics. Next slide, please. So for those who attended the IQ Consortium webinar on using Bayesian statistics for CMC, you may recall that Bayesian analysis is a far from new statistical tool, and that it's an alternative approach to classical frequentist statistics. There is no contradiction to frequentist statistics. The Bayesian method simply interprets the data in an inverse manner, while also including prior knowledge into the equation. As such, we consider Bayesian statistics to be ideally suited for the application of prior knowledge in predictive stability modeling. Next slide. In accelerating a product development program, one of the main challenges is the impact of small data sets, due to few batches of product having been manufactured. It is therefore desirable to include applicable prior knowledge into a stability model. The Bayesian method avoids the potential bias introduced by pooling data and the need for ANCOVA analyses to demonstrate the poolability of data as described in Q1E. Therefore, the precision of inferred statistics is improved. And we conclude that statistical analysis of stability data from small data sets has clear advantages by introducing prior knowledge data using the Bayesian statistics to mitigate the effects of small product data sets, thereby improving the accuracy of the statistics and any inferred predictions for product stability and shelf life. However, the challenge is in justifying the prior, or in other words, how transferable is your prior knowledge information to different molecule? Next slide. So at Amgen, we have tested Bayesian statistics against classical frequentist statistics and drug substance stability data for high molecular weight species at the initial MA submission for a standard IgG product were analyzed using frequentist statistics to establish tolerance intervals over time. For Bayesian analysis, the data were reduced to better reflect an accelerated product in development. The number of lots were reduced to 7, and the time reached in the stability program was no more than 24 months. Prior knowledge stability data were taken from 4 like molecules to include in the Bayesian calculation of tolerance intervals. Next slide. The comparison shows similar regression lines and tolerance intervals for the Bayesian approach. They use a reduced product data set supplemented by prior knowledge, compared to the frequentist approach using the full product data set. The tolerance bounds were notably similar when the Bayesian analysis was extrapolated twofold from 24 months to 48 months. This test indicates that the Bayesian approach would infer a similar shelf life for a product that has a reduced data set when supplemented with prior knowledge data. Next slide. Okay. We next use the reduced data set to test the effect of adding prior knowledge using the Bayesian approach. Clearly, in the absence of prior knowledge and with a reduced data set, the probability distributions for each time point increases markedly over time. When prior knowledge data added into the Bayesian statistics, the spread of the distribution was reduced fourfold for this attribute. Using this approach, we can also titrate the amount of prior knowledge, and we expect Bayesian statistics to inform on how much prior knowledge is needed to supplement a given amount of product data to give an accurate inference of the product shelf life. Next slide, please. For a second case study, we looked at the Bayesian analysis for a liquid drug product acidic peaks by cation exchange chromatography. Again, the stability data set from a recent monoclonal antibody marketing application, referred to as MAP0, was reduced in lots and for the time point reached, and supplemented with prior knowledge stability data from the 4 selected like molecules. Note that 2 of the molecules in this case were in glass vials and 2 were in glass syringes, and that 2 were IgG1 molecules and 2 were IgG2. Some knowledge from analytical comparability demonstrates no impact on product quality between the 2 different container types that have the same composition. Similarly, our analyses also indicate no stability indicating quality attribute differences between IgG1 and IgG2. Next slide, please. Similar to the high molecular weight species data, the comparison of frequentist full dataset analysis versus a Bayesian reduced dataset supplemented with prior knowledge shows similar regression lines, and the tolerance intervals were not too dissimilar when the Bayesian analysis was extrapolated from 24 months to 36 months. Again, this test indicates that the Bayesian approach would infer a similar shelf life for a product that has a reduced data set when supplemented with prior knowledge data compared to analysis using a full data set with the long-term stability data out to the shelf life. Next slide, please. Similarly, we next use the reduced data set to test the effect of adding prior knowledge using the Bayesian approach. Again, in the absence of prior knowledge and with a reduced data set, the probability distributions for each time point increases markedly over time. When the prior knowledge data are added to the Bayesian statistics, the spread of the distributions was reduced 2.4-fold. In summary, we conclude that adding prior knowledge using the Bayesian statistics gives accurate predictions from small product-specific datasets by better reflecting the uncertainty behind a small sample size of product data against the true data distribution. Next slide, please. So to recap, use of prior knowledge is critically dependent on justifying the transfer of data across different molecule types, that is, justifying the prior. Here we see a list of considerations that we make to justify our prior knowledge. Inevitably, as products are optimized from clinical use to the market, differences from any initial platform are likely. But what differences actually matter to the product stability profile? Whereas the initial analyses used multiple criteria, our Bayesian analyses selected molecules only according to their product structural class, pharmaceutical form, and the stability profile. And the similar results we obtained suggest that many of the other criteria were not really meaningfully impactful on the stability profiles within the ranges that we were using. To further investigate this challenge, we see artificial intelligence deep learning as a powerful tool to identify the quality differences between products that are important, and hence those that are less important. And the attraction of AI is that the analyses are impartially objective, and input parameters are tested while developing the model, which also develops its own relationships between the input data. And of course, the AI model would be subject to ongoing verification with concurrent parallel long-term stability studies. Next slide, please. Indeed, AI deep learning in the analysis of stability data is a tool that we've been developing to evaluate prior knowledge stability data for accurate prediction of stability profile. And an early prototype model is illustrated here. This model was trained for the percent high molecular weight species using 7 IgG molecules and was cross-validated by a leave one out process. On the upper right-hand side is the output of using this AI tool for a set of input values from a new IgG molecule that's shown in blue against the prior knowledge data that is shown in gray. At the lower right, you should note the linearity observed between the predicted level of high molecular weight species against the experimental values. And this gives confidence in the accuracy of the predicted level of the high molecular weight species in the new IgG molecule at 24 months using the AI deep learning model. So this analysis measured the impacts of differences across the prior knowledge molecule had on the stability profile. And the parameters showing differences are then used to define the inputs of the model for its next iteration. So overall, we see huge potential for AI deep learning in stability analysis when integrated into a data lake. And this tool could then evaluate all stability data for all molecules and to identify the best molecules to act as the prior knowledge. Next slide, please. So I just mentioned about using a stability data lake, and this slide illustrates how we're using such a data lake to manage our stability data in a structured manner. So data from disparate systems are being consolidated into a form that's readable by both machine and human. And metadata tags link all the interrelated data to form a data fabric that can be analyzed and interpreted to generate stability technical reports and regulatory filing documents. So this structured content data management retains data integrity, and information blocks such as data table can be reused in different submissions by relinking from the original component data. In other words, the dossier components with the structured data are reusable and traceable according to their required outputs. Additionally, the use of analytics could provide real-time trending analysis of stability data and the ongoing verification of any predictive stability model as part of the company quality management system. So Amgen and other companies are now developing cloud-based data lakes with interactive portals to share data immediately and directly with agencies. And automation may be used to auto-populate stability tables and perform stability profile analysis with data integrity that's been preserved. And so with that, I hand you back to Laurie, who will then conclude this webinar session.
Laurie, you're on mute.
Sorry about that. So we have come full circle, and we've seen a lot of the exciting things of the future, as well as some of the current ways predictive stability is being used for biologics in the overall science and risk-based stability paradigm. I reference here an article published by our IQ Consortium work group on considerations for updates to ICH Q1 and Q5C stability guidelines. The ICH has recognized that these are now out of step with the more science and risk-based approaches and have determined that they will be updated. And so we have made some recommendations here. And the predictive stability that we've talked about today is included in that assessment. So what we have been advocating for is an update to the overall core guidance to incorporate more science and risk-based language consistent with other recent ICH quality guidances. And in addition, to have some specific annexes that cover risk assessments, stability related, and risk-based predictive stability, as well as post-approval changes. So that's it for the presentation, and now we can move on to the Q&A with our other presenters joining us and Brian moderating.
Thank you. Excellent presentation, everyone. We did have some questions that have come in throughout the course of the webinar. One of the ones, a common theme that I'm seeing in a lot of these questions, has to do with the different approaches that were discussed today and proposed. What would it take for regulators to have confidence in these new tools in analyzing stability's prior knowledge data, such as Bayesian statistics and artificial intelligence? And do we have any actual health authority feedback that's been received on these approaches that you could share with us?
So I think I'll pick up on that one. So this is a topic that I'm very interested in learning more about the regulator's perspective. And we have heard previously from Matt Swellin, the EMA, the regulators need to be exposed to and become familiar with the new strategies and how we're using them and how we justify them. At the moment, quality assessors just haven't seen the examples using Bayesian statistics or AI, but believe me, we are actively working on that throughout this year. So I can appreciate that it's hard for regulators to evaluate innovative strategies without actually seeing a full package that's being used to support them or how they're being applied. So they need that exposure to these approaches to be able to identify what they see as being the gaps. They also need changes in the guidance as well to allow some of these new approaches. So nevertheless, clinical assessors are increasingly familiar with the application of Bayesian statistics, and the general principles and challenges are very similar that really just boil down to justifying the prior.
Right. I can add to that, Andrew, maybe that there should be at least a little bit of optimism there. If we take that step back, I mean, who would imagine that the use of prior knowledge in general and some of the approaches that were talked about in the first part of the talk would be both deemed acceptable and in fact encouraged in some ways by the health authorities, as they have been, particularly with respect to accelerated type of pathways, the BTD or PRIME, and are even in draft DMA guidance. So there is encouragement out there. I think the big challenge which again requires a guidance update, is taking those few health authorities that are very encouraging and supportive and expanding that to, globally to rest of world health authorities, so that you're not left in the bind of having both a really traditional program as well as a statistically driven or predictive stability program.
Yeah, and of course the challenge is that we can't really wait for ICH to be updated. That's going to take quite a number of years, and it hasn't. We hope it'll be starting this year, but we have to wait and see. But there is a lot of encouragement, as you were saying, Laurie, because the EMA has its draft guidance for using certainly the prior knowledge approach for products under the PRIME regulatory pathway, and industry is advocating that these approaches are expanded further from just the restrictions of a PRIME procedure to more of the unmet medical need, or preferably what I just call medical need. And we hope that the FDA, because this all derived from a joint EMA-FDA workshop, and we're waiting for the FDA output, which we are expecting sometime this year as a part of the new map and part of the Q&A. But I have no insight as to exactly what's going to be in there from the FDA.
Excellent. So switching over, there's a number of questions that have come in around the prior knowledge approach. So one of them, and we'll just go through them one by one as much time as we have, is there a metric that's used to define similarity between the biologics that allow their use as a prior knowledge to predict the stability of a product?
Yeah, that's a good one, because what we've done at the moment has been more qualitative. We can, you can tabulate to compare the different characteristics that we're looking at between the products, and then it's just what data will we have, or what literature is there that could support that there may not be a difference. In terms of quantitation, it's a very early work that we're getting from using AI might be indicating a way that we could actually provide some quantitation on this similarity, but I can't really say anymore. I've only just seen a very, very early output that was very encouraging from AI. But I think it's a very interesting point, and be something that will be very valuable in refining the Bayesian approach.
All right. Another question around prior knowledge is, can you go into a little bit more detail about how prior knowledge would be presented in a regulatory environment where it involves previously marketed products or products that are in ongoing clinical trials?
Yeah, so we have had some scientific advice on that topic. I've mentioned earlier that about providing the comparison of the prior knowledge molecules in a tabular form. Doing this by cross-reference was not encouraged. They really want to, they're not talking about anything complicated in terms of comparability. They just wanted to see a comparison of the products in the tabular form. The other point that was emphasized frequently was the need to have a risk assessment for the approach. And we know that the agencies are very keen on knowing the limitations of any form of science and risk-based approach.
Mm-hmm. And that's actually a great question. It comes into how when you're using the prior knowledge impact, does that impact your product design or any development decisions or milestones that you're making on the product itself, or on anything that's being used in the prior knowledge space?
Yeah. So, I mean, there are many applications of the predictive stability modeling, and it goes right from molecule candidate selection all the way through the development and commercialization process and into post-approval. So, things like the selection of your model and the outputs and all the parameters of interest probably all be varying according to the use that you're having for the model. Perhaps that sort of part answers that question.
Okay. And then one last question. I think we have 1 minute left. And for all the unanswered questions, we will be trying to go through those and putting them together and putting them on the IQ site. But what are some best practices for choosing and validating the right model for your stability studies?
Okay, well, this is something that we are very much looking at and considering at the moment. It's really is a big topic and a very broad question. And again, it comes down to the purpose of the model that you're developing. And of course, the ability to have the prior knowledge that you can justify. So, before marketing application, the approach should be justified as appropriate for intended use. May not be fully validated until the marketing application. However, both the prior knowledge approach and the kinetic modeling approaches, they both will have ongoing verification of the models. And to me, that's a very key point. And the use of AI involves extensive validation of its software modeling as a part of its development and is following engineering industry standards. So when it comes, if you got a new structural moiety, you may not have the prior knowledge data set that can apply. So then, clearly you might want to go with a kinetic modeling approach, but you still got to, consider the regulatory environment. For a well-established product that's using a platform like a monoclonal, the use of prior knowledge or kinetic analysis may both be applicable, but again, depends on the regulatory environment at the time as well, as to which you see as being most useful. And as discussed, the ability to use prior knowledge is critically dependent on justifying that transfer of that knowledge.
Okay. And with that, I think we've hit the top of the hour. So I'd like to thank everyone for their attention and attendance today, as well as for the questions that were submitted. For those questions that we did not get to at the end of the webinar, as I mentioned, we will be going through these and trying to get a written response together. If there are any additional questions, please feel free to reach out to any of the speakers or to myself, and we'll try and get an answer to you as soon as possible. But thank you very much for attending today, and hopefully you'll be able to make our 2 future webinars next Thursday and the Thursday after. So with that, we're going to close out the webinar, and thank you very much.